So can you hear me? That works. Well, thanks, Bilal. It's really a pleasure to come here by Bilal's invitation because I've known Bilal since we were training together at Yale, and I have to say that I've admired him since basically the moment we met, and because your intelligence and your passion about neuroscience is so clear. So thank you for inviting me. Thank you all for having me here. I'm going to talk today about some of the work that my lab has done over the last few years, trying to understand how the brain balances actions and habits, and then how these processes can be affected by cocaine. And if you can't hear me, please just raise your hand there in the back. Okay, so generally speaking, my lab at Emory is really interested in understanding the long-term effects of cocaine and or stress hormone exposure. And we focus quite a bit on adolescents because a lot of epidemiological evidence will suggest that if you are an adolescent and you're exposed to cocaine, you have worse long-term outcomes than individuals who are exposed as adults. So for example, you're less likely to seek treatment for drug use disorders at any point in your lifespan relative to individuals first exposed to cocaine as adults. And so there seems to be some effect of cocaine and other pathological insults like stress on decision-making processes. So we're going to talk about that today, and within the brain we're going to focus on the prefrontal cortex, and in particular on the orbital prefrontal cortex, or OPFC. So I'm highlighting it here in purple on a coronal section from the mouse brain atlas. And we're interested in the orbital cortex in my lab for a lot of reasons. For example, it's hypoactive following cocaine exposure. This is in individuals who have had cocaine and they're now off drug. Even though they're now drug abstinent, they're still showing hypoactivity of the orbital prefrontal cortex. This region would otherwise be really important for allowing us to engage in inhibitory control and to choose the best behaviors amongst many. So if you're a rodent and I give you cocaine, we see the loss of dendritic spines, or the primary sites of synaptic contacts within the orbital cortex. And this is very much unlike many other brain regions where cocaine actually causes dendritic spine proliferation. If you're an adolescent mouse and I give you cocaine, we actually see the loss of your dendritic trees within the orbital cortex. And so we see pretty gross changes in the neuroanatomical composition of the orbital cortex in individuals and animals exposed to cocaine. And these changes are long-lasting. And so we think about them quite a bit in the context of how cocaine may have long-lasting behavioral effects. So we'll focus today on actions and habits, and I'll define these terms in a moment. And then we're going to hone in on a potential molecular signaling mechanism, brain-derived neurotrophic factor, acting through its high-affinity receptor TrkB. And then finally, we're going to try to take what we've learned about how the orbital cortex balances actions and habits, how BDNF may be involved in this process, and we're going to try to block habits that result from cocaine exposure. And then in my last 3 slides, I'd like to show you some new data that we've been collecting that the data have taken us out of our favorite brain region, the orbital cortex, and unexpectedly into the prelimbic prefrontal cortex, which is also, it turns out, involved in action outcome decision making. All right, so we're really excited in my lab about the orbital cortex because it's anatomically poised to really contribute to complex decision making. So if we look at anterograde tracing studies from my lab and from many other labs, if we simply infuse an anterograde tracer within the orbital cortex in the mouse, we can see really nice terminal projection patterns in the dorsal striatum and in the amygdala. And this is really exciting because these 2 regions are important for goal-directed action as opposed to habits. All right, okay, so I've mentioned this action versus habit thing once or twice before, so let's pause and define these terms. So a goal-directed action is exactly what it sounds like. It's engaging in some sort of behavior with the expectation that that behavior will be reinforced. So you came here today with the expectation that your action would be reinforced with maybe a decent talk. Often I get laughs there. There you go. By contrast, habits are different. They're stimulus elicited. So these are typically conceptualized as familiar behaviors that we've engaged in so many times that they're no longer linked to the action outcome contingency, but instead are stimulus elicited. So one example from my life has to do with the fact that for many years I wore glasses. So I don't wear glasses so much anymore, but as those of you who do wear glasses know, when the glasses fall down on the bridge of your nose, you have a hard time seeing because you're not looking through the lens, and so you bring your hand up to adjust your glasses, right? So again, I don't wear glasses so much anymore. However, when I have a hard time seeing, such as the lights in the room dim for a lecture, I bring my hand up like this, okay? So this is a classical habit. There's some sort of stimulus, that is that I have a hard time seeing, and there's some sort of motor behavior, this. So you can imagine that habits in many cases may be innocuous, so this makes me look kind of weird, but it's not hurting anybody. However, habits that go awry, so in the case of, for example, drug-seeking habits that we have a hard time inhibiting, these kinds of habits may contribute to addiction. And indeed, one of the prominent models of addiction argues that addiction can begin with occasional drug use. It can then proceed to habitual drug use. And then in some individuals, according to some environmental or genetic circumstances, that habitual drug use can then proceed to compulsive use and relapse. And so my lab is really interested in understanding the mechanisms by which habits form and the mechanisms by which we modify our behaviors when our habits are not advantageous. And so to do this, we use our operant conditioning chambers here. We have a bunch of these chambers in the lab. And basically what you're seeing is that our mouse can rear up on, or can explore the operant conditioning chamber. This animal is rearing up against a nose poke panel. This nose poke panel has 3 nose poke recesses, and when he completes the response requirement, he gets reinforced with a food pellet that's delivered through the dispenser up here, down the tube, and into the magazine. So there's a mouse waiting for his food pellet. So in the task that we're going to talk about today, if we zoom in on the nose poke panel, we're first going to train the animals to respond on 2 nose poke recesses, so the outermost and the innermost nose poke recess. And we're going to ask the animals to respond equally on both recess, and that very technical graphic that I just showed you there serves to remind me to tell you, thank you for laughing, to tell you that when we train the animals, we're training them to have equivalent experience with both nose poke apertures. They're being reinforced on a fixed ratio 1 schedule of reinforcement, which simply means that every nose poke is reinforced, and the experience with each nose poke is controlled such that 30 pellets are associated with each nose poke aperture. So over the course of training, which takes about a week, we can generate animals that are A, very proficient in responding on both the left and the right nose poke aperture, but B, have equivalent experience with this reinforcement. And this is really important because it allows us to then ask the mice how they're making decisions in their environment. So we're going to use what's called an instrumental contingency degradation procedure, which is a fancy term for something pretty simple, actually. So as a reminder, our mice have learned now that they can get 30 reinforcers by nose poking on the left and the right nose poke aperture for a total of 60 pellets across the session. And now we're going to play a little bit of a trick on the mouse. So we bring him into the conditioning chamber on one day, and we've occluded one of the nose poke recesses. He has access to this one, let's say. And now his responding is reinforced, but only on one nose poke. So he gets pellets just like he always did on this nose poke recess. He goes back to the colony. We bring him into to the chambers the next day, and now we're going to degrade this contingency, this action outcome contingency. And to do this, we're going to deliver food pellets for free. So the animal can nose poke as little or as much as he wants, and rather than being reinforced, food pellets are going to be delivered into that magazine at the same rate that he earned them yesterday. So if you let me anthropomorphize just for a minute, if you're a mouse and you're in this task, what you should be thinking is, wait a minute, it doesn't make sense for me to nose poke over here because my nose poking no longer results in reinforcement. So we can ask the mouse if he has learned this and if he's utilizing this new information by simply bringing him back to the conditioning chambers the next day for a probe test conducted in extinction. This is a very brief test. We stick him in for 5 minutes, and we're simply asking the mouse where he wants to spend his time. So a typical mouse with moderate training, and you'll see a few examples of this in a moment, will preferentially engage the response associated with the intact or non-degraded contingency, and he'll, relatively speaking, he'll ignore the degraded response outcome contingency. So he's modifying his behavior. Previously, he went into the chambers and responded equally on both nose poke apertures. Now he's modified his behavior, so he's preferentially engaging a behavior that's likely to be reinforced. So by contrast, if we generate mice that have had a history of lots and lots and lots of training, we can engender habits, just like me and my glasses adjusting behavior. And we have a mouse that will run into the conditioning chambers, and he'll bang away on both nose poke recesses, just like he always did, because he's developed this left-right nose poke habit. Okay, so we also appreciate in the literature that if you expose mice or rats to lots and lots of cocaine or amphetamine or alcohol, we can engender habit-based behavior that forms before it should. So these are premature habits. And then additionally, if we ablate or otherwise inactivate the orbital frontal cortex, we can also cause habit behavior that fails to be modified. And we think it's because animals are not able to learn new response outcome contingencies, so they defer back to what is familiar. So this paper by Gremmel and Costa came out in 2013, and it was really impactful and continues to be impactful for the field because they show, using several inactivation techniques, that in mice, the orbital cortex is necessary for learning about response outcome contingencies, just as in monkeys. Jane Taylor and I had a paper out the same year showing using one inactivation technique, the same thing, but that was nice because there was concordance. But additionally, we were really interested in understanding potential molecular mechanisms. And so one sort of obvious mechanism, to us anyway, was BDNF. So BDNF, or brain-derived neurotrophic factor, is a neurotrophic factor that's expressed throughout prenatal and postnatal life. You guys probably have heard about it in the context of, say, axon guidance during early development. But BDNF is also really important for the stability of dendrites and dendritic spines in adulthood. And so to ask whether BDNF was important for the functions of the orbital cortex, we simply took mice that were homozygous for a flox BDNF gene. We infused bilaterally into the orbital cortex an AAV Cre to reduce BDNF expression. And then we trained the mice to perform the nose poke responses, and then we asked them if they can modify their behavior based on changing response outcome contingencies. And what I think is pretty obvious is that the mice with the knockdown could acquire the nose poke responses, but they generate lower response rates. That's pretty typical of orbital lesions across lots of species. But what was more exciting for us was that when we modified the response outcome contingencies, our control mice modified their behavior. So instead of responding equivalently on both nose poke apertures, they prefer the nose poke that was likely to be reinforced. By contrast, mice that have BDNF knockdown are unable to modify their behaviors. So in 2011, as Bilal mentioned, I had moved to Emory, and I had this great student who was really interested in understanding this effect further. So this is Kelsey Zimmerman here. And so the first thing that I asked Kelsey to do was to replicate my original experiment. So at Emory, we're obviously geographically located in a different part of the country. We use different mice. So these mice are bred on a bulb C as opposed to B6 background. And then we're also using a lenti Cre from the Emory core as opposed to an AAV Cre from Ralph de Leon. So there are a lot of differences in the experimental details here. Nevertheless, I hope you can appreciate that Kelsey pretty readily replicated my finding. So first, if she knocked down BDNF within the orbital cortex, our mice could learn to perform the nose poke responses, but the knockdown mice have lower overall response rates. More importantly, when we modify the response outcome contingencies, our mice with BDNF knockdown are unable to keep up. And so they defer back to a familiar approach. And so this tells us that BDNF within the orbital cortex is really important for the functions of the orbital cortex in terms of modifying behavior or engaging in behavioral flexibility when response outcome contingencies change. And in the absence of that BDNF, we might instead defer back to a familiar habit. So Kelsey really wanted to ask which circuits might be important for these kinds of behaviors, and then can we use what we've learned to block habits that may emerge due to, for example, cocaine or stress hormone exposure. And so to take on the first question, we used what's called a disconnection strategy. And so Kelsey and another student of mine, Liz Pitts, were really interested in understanding whether the BDNF expressing projections from the orbital cortex to the amygdala were important for response outcome learning and memory. And so again, to test this strategy or to test this potential connection, they used a disconnection design. And what we're doing is we're taking advantage of the strong evidence that in the mouse, the ventrolateral orbital cortical projections to the amygdala are ipsilateral. And so what that means is that the left orbital cortex is talking to the left amygdala, and the right orbital cortex is talking to the right amygdala. And so what we can do then is reduce BDNF within one hemisphere of the orbital cortex, and in this case, chemogenetically silence the BLA and the contralateral hemisphere. And when we do this, we're causing a disconnection because our BDNF deficient orbital neurons are trying to talk to a healthy amygdala, and our healthy orbital neurons are trying to talk to a silenced amygdala. So in these designs, you always have, of course, have viral vectors that are expressing fluorophores as your control, but additionally, you can infuse your Cre-expressing viral vectors and your DREADs within one hemisphere. And in this case, you're knocking out one connection, so knocking out, let's say, the left orbital cortex talking to the left amygdala, but you have your right network intact. So we also went into these experiments with the hypothesis that BDNF-dependent interactions between the orbital cortex and the amygdala would be particularly impactful during the consolidation or the retention of new learning and memory. So we took our mice through all of the training and testing that I've described to you so far, but we didn't inactivate the amygdala until immediately following the contingency degradation day. Just as a reminder, this is the day that the mice got free pellets. So this is the day they should be learning something new, and that is, well, wait a minute, this nose poke response over here doesn't matter anymore. I don't need to generate this. It doesn't yield food reinforcement. And so immediately after that session, we pull the mice out of the conditioning chambers and we inactivate the BLA, the basolateral amygdala. We bring them back to the testing chambers the next day for a probe test. And again, we're simply asking the mice where they want to spend their time. And so what you can see is that in both male and female mice, our typical animal, our control viral vector expressing animals can preferentially engage the response that's likely to be reinforced and neglect the response that's unlikely to be reinforced. The same is true of the mice that have one connection spared. By contrast, if we have our contralateral groups, we see that these mice are unable to update their response strategies, so they defer back to what is familiar, and that is to engage both responses equally. So what this tells us is that BDNF-dependent interactions between the orbital cortex and the basolateral amygdala are really important for laying down this new response outcome memory, which then allows you to engage in behavioral flexibility later on. Any questions so far? Okay, so we were really excited, that this timing worked, right? So this tells us something really critical. This tells us what phase of learning is being affected by at least the deficient BDNF or the deficient orbital amygdala interactions that we're impacting when we knock down BDNF. And so we capitalized on this finding by trying to block habit-based behavior that resulted from bilateral BDNF knockdown. And so now we're going back to a scenario where we're reducing BDNF bilaterally within the orbital cortex of our mice. We're training them to perform 2 nose poke responses, and then we modify the likelihood that one response will be reinforced. Immediately following that session, we pull the animals out of the conditioning chambers, and half of the mice receive an experimental novel compound called 7,8-DHF. So 7,8-DHF putatively has agonistic activities at track B, which is the high affinity receptor for BDNF. And after they've had this injection, they go back to the colony, and then we bring them back to the chambers the next day when they're drug-free, and we ask the mice where they want to spend their time. So as I showed you a few slides ago, BDNF knockdown causes this deferral to habit-based behavior. However, if we intervene with this track B agonist, we can rescue the decision-making strategies. Now, Kelsey wanted to ask whether this track B agonist could block habit-based behavior that occurs under more naturalistic circumstances. And so she took intact, typical mice, and she trained them every day for a month using escalating random interval schedules of reinforcement. And the reason this is important is because, as you can see, it generates really high rates of responding that cause habit-based behavior in our mice. Again, just like me with this glasses behavior that I engage in because I've done it like a million times before, our mice generate habit-based behavior because they've nose poked a million times before. So again, immediately after this contingency degradation procedure, Kelsey pulled the mice out of the conditioning chambers. She injected half with vehicle, half with 7,8-DHF, and she brought them back to the chambers the next day, drug-free, and asked them where they wanted to spend their time. And so our control mice develop habits as expected. By contrast, our mice that were given 7,8-DHF are able to really nicely, flexibly modify their behavior, suggesting, suggesting that this track B agonist is facilitating the functions of the orbital cortex in this task. So I'm not going to show you the data for time, but just as a side note, since this initial paper came out showing that 7,8-DHF causes phosphorylation of TrkB, this finding has been questioned quite a bit. And so in this experiment, we replicated this whole experiment, but using a 2x2 design, where some mice received both 7,8-DHF, the TrkB agonist, but they were pre-treated with Anna-12, which is a TrkB antagonist. And that was able to block the effects of the 7,8-DHF, the putative TrkB agonist. So this is all just to say that although 7,8-DHF, if you are familiar with it, it may indeed have unintended off-target effects. We think that at least this effect that I'm showing you here can be attributed to its activity at TrkB. Okay, so summarized here is a lot of work, a couple of years of a PhD thesis, but ultimately what we want to do is get to the point where we can develop new strategies to block habit-based behavior that results from, say, cocaine or stressor exposure. And we were particularly interested in adolescent cocaine exposure. And I had sort of alluded to this earlier, but the reason is that when individuals are exposed to cocaine during adolescence, there are long-term impacts on decision-making processes. And additionally, we know from rodent studies, you have a long-term impact on the anatomy of the orbital cortex. So for example, if we expose mice to cocaine from postnatal day 31 to 35, this is a pretty brief period that would correspond to early adolescence, or ages like 12, 13, 14 in humans, and then allow the animals to be drug-free for 3 weeks and then look at their orbital neurons, we see that the basilar dendrites are atrophied in layer 5, and then additionally these neurons are less complex, which is what you're seeing in a shoal analysis here. On top of all of this, we see a loss of dendritic spines in the same brain region. And so we're seeing, again, these long-term consequences, this long-term simplification of orbital neurons, and we think that it may be associated with some of the decision-making deficits I'm going to talk about on the next slide. And this, I'm sort of jumping my own gun here, just to confirm that this same cocaine exposure procedure can cause habit-based behavior in our mice. So again, if we expose cocaine to mice during adolescence and we use the exact same task that I've been discussing so far in adulthood, we find that our cocaine-exposed mice are less behaviorally flexible, they're less able to modify their behaviors when action outcome contingencies change. And I'll show you an example of this in a moment. But really, the end goal here is to correct these habits, right? To improve the animal's ability to engage in behavioral flexibility. And so my student Liz sort of combined these two projects, and she asked whether we could block cocaine-induced habits by this TrkB agonist, 7,8-DHF. And so she used exactly the same timing that I mentioned previously. So we're giving mice cocaine for 5 days from P31 to P35. We're letting them grow up without any more drug exposure. We're taking them through exactly the same conditioning procedure that I've discussed so far. And just as I promised you, our mice that have had a history of cocaine exposure develop habit biases as adults. So they're less able to engage in behavioral flexibility based on action-consequence associations. However, if Liz pairs 7,8-DHF, this TrkB agonist, with the opportunity for new learning, she can rescue the animal's decision-making. So the animals can come into the operant conditioning chambers and preferentially engage a response that's likely to be reinforced as opposed to one that's unlikely to be reinforced. So as I mentioned or alluded to earlier, whether 7,8-DHF has TrkB agonist properties has been questioned. And so Liz did her homework here, and she systemically administered this TrkB agonist and took brains from the mice at the same time point that we think this new memory is being laid down. And when she extracts the orbital versus medial prefrontal cortical tissue, she sees a main effect of drug on TrkB phosphorylation, but I think any of us could look at this graph and recognize that it's likely being driven by the signal in the orbital cortex. And so why this TrkB agonist would preferentially penetrate the orbital cortex, we don't know yet, but it may be something that is advantageous to us in the future. And so, so that was really exciting, but what is sort of less exciting about 7,8-DHF, and BDNF track B approaches in general is that BDNF track B is really important for pain systems. And so giving a systemic administration of a track B agonist would be very much expected to heighten pain sensitivity. So obviously this is a major drawback if we're thinking about clinical applications, right? It's very unlikely that this would be really translatable. And so Liz, to her credit, started to think a little bit outside of the box here and tried to find drugs that would really stimulate BDNF track B systems preferentially in the cortex as opposed to other parts of the nervous system. And so Liz stumbled upon MDMA. And yes, it's that MDMA that you're thinking of. So it is, we're talking about MDMA, the club drug, MDMA. Turns out at very low doses, so doses that are not reinforcing to us or to mice, can cause BDNF stimulation in the cortex, or BDNF release, excuse me. And so Liz first wanted to ask whether MDMA could block cocaine-induced habits, and indeed she was successful. So here we can see that in a separate cohort of mice, if the animals were exposed to cocaine as adolescents, they have a hard time adjusting their behaviors in adulthood. However, if she pairs low-dose MDMA with this opportunity for new learning, the animals can later on can engage in behavioral flexibility and preferentially engage the action that's most likely to be reinforced. So again, Liz did her homework here. She systemically injected MDMA, took the brains, and confirmed that indeed we see an elevation in mature BDNF within the cortex, as you can see right there. Additionally, we see an elevation in phosphorylation of ERK, which is one of the downstream substrates of track B. And then confirming other studies, we actually see a loss of BDNF within the dorsal striatum, and interestingly, no effects within the medial prefrontal cortex or amygdala. And so I would say, while this profile is really advantageous to us, one really important future question will be to understand why MDMA has such different effects throughout different brain regions on neurotrophic systems. So in all of the work that I've shown you so far, we've been reducing BDNF selectively within the orbital cortex using viral vector strategies. And then I've been stimulating track B using systemic approaches, so 7,8-DHF or MDMA. And so these data have all really nicely come together to argue that BDNF within the orbital cortex is really, really important for our ability to flexibly adjust our behavior based on action-consequence associations. What it doesn't tell us is where track B binding is important. So again, track B is a high affinity receptor for BDNF. So this might seem like a silly question to you, like if BDNF in the orbital cortex is important, obviously track B in the orbital cortex is really important, but that's not necessarily the case because BDNF is subject to anterograde and retrograde transport. So it may be that when we knock down BDNF within the orbital cortex, we deprive, let's say, the amygdala of BDNF, and it's really binding within the amygdala that's important. And so we wanted to ascertain where track B binding in the brain was important, and to do this, we teamed with Carrie Ressler, who had created a lentiviral vector that overexpresses a truncated form of track B. So track B comes in two flavors. One is the full-length isoform that has the intracellular tail, and one is a truncated isoform that doesn't. And so when track B binds, it has to dimerize and autophosphorylate. So to be an active track B receptor, you need both of those full-length tails. So here, and then, for example, you can phosphorylate ERK, the same kinase I mentioned on the previous slide. However, if you have only truncated isoforms or one truncated isoform, you are inactive. And so by overexpressing a truncated form of track B, we're essentially competing with endogenous full-length track B for BDNF. And so again, this is our viral vector here. That's our friend Carrie there. And so for this experiment, Liz actually infused 2 volumes of the truncated track B viral vector. So some mice received the 0.5 microliter volume, other mice received the 1.5 microliter volume, and what you're seeing here is histological representations of the infusion spread. And what I hope you're thinking is, Shannon, that looks exactly the same. It does. When we quantitatively measure the viral vector spread, we actually don't see any differences based on the volume. And that's important for a reason I'll show you in a minute, but it's also not not surprising. So if any of you have worked with lentiviral vectors, you know that they're really, really sticky. So they tend to stay where you put them, which is really handy if you're working in a very small mouse brain like us. And so what we're really intending to do by infusing 2 volumes of this viral vector was not to generate a larger spread, but to generate a greater degree of knockdown. And so as a surrogate for this approach, as a surrogate for this measure, one of the things that we did was we measured or quantified the HA tag on the viral vector. And indeed, what you're seeing is in what we're calling the half or the low dose viral vector, we see a nice signal, but it's much, much, much stronger in the sort of quote unquote high dose viral vector. And so all I'm trying to get at is that we took some principles from pharmacology where you might expect dose-dependent responses, and we applied them to viral vector strategies where we would expect here to see dose-dependent effects. And that was indeed the case. So when Liz tested her animals, you can see that all of the mice, again, with bilateral infusions of the orbital cortex, could acquire the nose poke responses, but our mice with a full dose, quote unquote, of the truncated tract B were unable to generate as robust response rates, as is typical of orbital damage. And then more importantly, when we modified these response outcome contingencies, our mice with the quote unquote full dose of the truncated tract B viral vector were unable to keep up. So this looks a lot like BDNF knockdown, and it suggests that BDNF knockdown binding to local tract B in the orbital cortex is at least is somewhat or rather quite important for engaging in response outcome contingencies or behavioral flexibility. And I'm sort of hedging there because I think that it's very likely that BDNF tract B systems in other brain regions may also be implicated in this kind of behavioral flexibility. But certainly our data would suggest that the orbital cortex is a primary place where BDNF tract B is really important, again, for behaving based on action consequence contingencies. And then just for the interest of time, I'm not going to show you the data, but we were able to block the behavioral effects of MDMA. And so again, this suggests that MDMA requires tract B within the orbital cortex to have these pro-cognitive effects. So if we can just pause for 1 minute. What I showed you today so far was that BDNF tract B within the orbital cortex is really important for selecting actions based on anticipated consequences. Okay. We think this is occurring via local orbital tract B binding and interactions with the amygdala. And then conversely, cocaine weakens memory for consequences. And this occurs, I should mention, in both rodents, as I showed you today, but also humans. So Karen Ersche had a paper in Science a couple of years ago now showing, using a contingency degradation procedure, just like the one that I've been talking about, that humans with a history of cocaine abuse are more biased towards making stimulus-dependent, habit-based responses in an operant conditioning task as opposed to, quote-unquote, goal-directed action outcome decisions. And so the findings I'm showing you here have been directly translated into human experiments. And then I showed you today that 7,8-DHF can both, and MDMA can both restore action and consequence decision making. And then just as a shameless plug, we also showed that 7,8-DHF can restore action consequence decision making following stress hormone exposure. Okay, so if we take a step back, what we think the orbital cortex is doing in general is that some sort of learning-related plasticity within this brain region is enabling us to retain action-consequence memories and thus engage in the most optimal behaviors later on. And I say retention here because of this study, for example, that has parsed out these different phases of learning and memory using inactivation techniques. So if any of you in the room are familiar with all of this action consequence, action outcome literature, you might have been wondering this whole time, why am I not talking about the prelimbic cortex, which is situated here in yellow? So the reason you're thinking about that is maybe you're thinking about the work of Bernard Belain that has shown since the late 90s that the prelimbic cortex is also important for action consequence decision making. So the prelimbic cortex appears to be important for the acquisition of those associations and also the consolidation of those associations. And when I was just starting my lab, we tried really, really, really hard to determine, well, to find a role for BDNF in these processes within the prelimbic cortex. And despite a lot of our efforts, we really couldn't. So it doesn't seem like BDNF is important for the function of the prelimbic cortex in acquiring action outcome associations. However, during this same time, I started to become interested in PI3 kinase. And in my last 3 slides, I want to show you some of the new data that we've been generating showing that PI3 kinase may actually be important for the functions of the prelimbic cortex. So PI3 kinase is a signaling complex that helps us transduce signals from the cell surface to the AKT-mTOR pathway. And one of the reasons that it sort of came onto my radar is that it's upregulated by cocaine, and the effects are really long-lasting. And so again, if you expose an animal to cocaine and euthanize it 3 weeks later, you can see an upregulation of AKT-mTOR phosphorylation within the medial prefrontal cortex. So it's involved in synaptogenesis and dendritic spinogenesis, so somewhat unlike the BDNF track B pathway, which might be more associated with dendritic spine stabilization. It's composed of P85 and P110 subunits, of which there are 8. So this really is a signaling complex, and there are lots of different bits and pieces that we can manipulate. What we care about in the context of again, these next few slides is P110 beta. So P110 beta is a catalytic subunit, and it's downstream of G protein coupled receptors. And I was interested in P110 beta, or became interested, thanks to my colleague, Gary Bissell. So Gary studies Fragile X syndrome, and he contacted me in 2012, and was like, hey, Shannon, we've got these fMR1 knockout mice, which are, as you guys probably know, a model for Fragile X syndrome, and we find this crazy thing, P110 beta is way, way, way upregulated. I mean, there's way too much of it. And so we think that potentially if we could reduce P110 beta, knock it down, we might be able to normalize some of the aberrant decision making that these mice engage in putatively. Gary doesn't do much behavioral work. And so he said, we've got the mice, we've got the virus, you've got the boxes, let's get together. And I said, okay. And so we stuck this viral vector that reduces P110 beta into the prefrontal cortex of our Fragile X model mice. So we just flooded the prefrontal cortex with this viral vector. And we were able to indeed rescue some of their decision-making impairments. And if you're interested, this is the paper. So all this time, we're thinking about, well, Gary's thinking about Fragile X. I'm thinking about cocaine, right? And I'm thinking about cocaine because of this paper that came out way back in 2002. And this paper showed that if you infuse into the ventricles a pan-PI3 kinase inhibitor, you can block the expression of cocaine-induced locomotor sensitization. And so the reason this was really, really exciting, even though it lacked brain region specificity, for example, was that the investigators were able to block this plasticity that occurred as a result of cocaine. So the animals are already way experienced with cocaine, lots and lots of drug, and they were able to block sensitization. And so this is a lot like people, right? So if you're an individual who has developed cocaine addiction, it's then that you're seeking treatment. You're not coming in before the cocaine exposure starts to impede and impair your life activities. And so we were wondering whether we could inhibit the P110 beta in particular to block some of the long-term consequences of cocaine. So we raised a little money from Children's, and I actually didn't know this, but we're in a Children's building right now. It says Children's, so that's very, very fitting. So we raised a little bit of money, and off we went. So first we needed to confirm that P110 beta was nicely poised within the prefrontal cortex throughout adolescent development to potentially regulate prefrontal cortical dependent behaviors. So we can see really nice expression patterns, really robust expression patterns throughout the medial and lateral orbital prefrontal cortex throughout postnatal development. And then we infused the viral vectors selectively in the medial prefrontal cortex to knock down P110 beta. And I have to credit Nina Gross for making these viral vectors. And so in one of the experiments we did, we really wanted to sort of draw inspiration from that 2002 paper and block some of the behavioral consequences of cocaine exposure. And so in this experiment, we implanted mice with indwelling jugular catheters that allow the animals to, instead of self-administer food in the conditioning chambers, actually self-administer cocaine. So instead of going into the conditioning chambers and nose poking on 2 recesses for food, they instead nose poked on a center recess for an infusion of a cocaine reinforcer given directly. So what you can see is that the mice can acquire the correct nose poke response. They neglect the nose poke response that is unreinforced over the course of several sessions. We then take them through extinction, which is a pretty common technique used in my field to sort of mimic therapy for better or worse. And then after all of this, only after all of this exposure do we infuse the viral vectors, which reduce P110 beta in the medial prefrontal cortex. Then we brought the mice back for a reinstatement test. And so here we're simply playing the mice cocaine-associated cues, and we're asking our mice, does this make you want to respond? And our scrambled control mice generate lots of responses, as you might expect. By contrast, this is pretty nicely mitigated by the selective knockdown of P110 beta. So it was pretty hard not to get excited about these data, so we ran a whole bunch of other experiments. And to summarize here, we can block the reinstatement of cocaine seeking under multiple conditions, including context-induced reinstatement. We can also block the expression of locomotor sensitization. So this fully recapitulates that 2002 paper that used a pan-PI3 kinase inhibitor in the ventricles, right? So we're honing in quite a bit relative to that first finding. And we also mitigate cocaine-induced elevations in phospho-mTOR, which is what you would expect based on the signaling. And so, of course, you're wondering, what about our habits? And so we took the mice through exactly the same procedure that I've been explaining for the last, X number of minutes now. And we, again, exposed the animals to cocaine before we infused the viral vector. So these animals have had a lot of cocaine exposure. In this case, we used a procedure from Schoenbaum and Setlow. This is the first evidence in rodents that cocaine causes habits, again, as in people. And then we have a washout period, and then we infuse the viral vector. And then finally, we train the mice to respond for food, and we take them through the contingency degradation procedure. So you can see during acquisition, mice with a history of cocaine exposure generate higher response rates. This is pretty characteristic and had been seen in previous studies. What's more important is that our mice that had a history of cocaine exposure generate habit-based behavior just like you would expect. By contrast, we can mitigate those habits and engender behavioral flexibility by knocking down P110 beta within the medial prefrontal cortex. These same data can be shown in a slightly different way by simply generating a response preference ratio. So in this case, you have the non-degraded response rate over the degraded. 1 is a habit, it's no preference. And so our cocaine-exposed mice are generating habit-based behavior like we would expect, and we can rescue that pretty nicely with the P110 beta inhibitory viral vector. And so to summarize, this catalytic subunit P110 beta is expressed really nicely throughout the postnatal prefrontal cortex. And in the absence of cocaine, it really doesn't do very much. Doesn't do much at all. And so, for example, this is a whole list of behavioral assays that mice that were cocaine naive but had the viral vector were tested, and we really didn't see any phenotypes. However, when we layer on cocaine, we start to see that cue-induced reinstatement, for example, can be blocked. This is the field's primary test for relapse-like behavior. We can block locomotor sensitization and cocaine-induced habits. And so we're trying to, just in general in my lab, we're trying to hone in on these maybe more subtle therapeutic approaches, such as a single PI3 kinase subunit, or maybe low-dose MDMA paired with the opportunity for new learning to try to help facilitate behavioral flexibility in individuals that are struggling with drug use disorders. And so in terms of our next steps, some of the things that we're thinking about a lot are whether habits are associated with or preceding addiction-like behaviors. This may seem like an unexciting question, but it's actually really important because it could shed light onto whether habits, for example, could be predictive of addiction-like behavior, or whether looking at habit biases, or whether it might be too late at that point. And because habit biases can be tested in humans, this has direct translational appeal. So one of the reasons we care a lot about BDNF and PI3 kinase is that they affect neuron structure, as I mentioned throughout my talk, and we think that this is really important for the behavioral consequences of these manipulations. And the reason we think this is, for example, if you simply look at typical healthy mice and you count dendritic spine densities, and in particular mushroom-shaped spines, these are the spines that are likely to contain synapses within the orbital cortex, you get a really nice predictor of how the animals will respond in our operant conditioning chambers. So will they engender action outcome-based decision-making strategies, or will they rely on habits? And so you can see here a correlation between mushroom-shaped spines and that response preference, where anything greater than 1 is a goal-directed strategy and anything at 1 is a habit-based strategy. And we think that the effects of these compounds on dendritic spines may be brain region specific, and also, more importantly, the effects of dendritic spine plasticity on behavior is going to be highly brain region specific. So, for example, within the prelimbic cortex, we just published that pruning of prelimbic cortical dendritic spines was really important for enabling the animals to generate action outcome decision making. And I need to thank Haley here, who actually helped quite a bit with this project. So thank you, Haley. And then finally, we're trying to understand how all of these molecules and these circuits are actually coordinating. So why would the prelimbic cortex rely more on PI3 kinase than BDNF, for example, in acquiring action outcome contingencies? And so these are questions that we're trying to tackle using protein quantification and direct manipulation of specific circuits and specific proteins. So I'm going to stop there. I would like to thank my team at Emory. We have a lot of great graduate students in the Emory Neuroscience program. Some of you may actually be in the Emory Neuroscience program, and so you know. This is the team that was largely in place when these studies were conducted. We have other folks who preceded and came later. And then thanks to Carrie Ressler, Gary Bassell for essential reagents and collaboration. Thank you also to you, and I'd be happy to answer any questions.
We have time for a few questions, Matt.
Hey.
I'm curious if you've thought at all about sex differences in this, both because one, there's differences in sort of susceptibility to some of the habit
Yep.
but also that this is all happening at the same time and using some of the same systems of sexual differentiation processes.
Great. Yeah, so we have thought about sex differences in a couple of contexts. So firstly, we have looked at individual differences in cocaine self-administration during adolescence, and then how the animals make decisions in adulthood. And we find that in females that are resilient to cocaine, so in other words, females that can develop really behaviorally flexible strategies in adulthood even though they've had cocaine, females generate larger dendritic spines heads within the orbital cortex. So they're able to recruit, it seems, these neurons to help them be resilient to the drug, whereas we just don't see this in males. And we also see that females, we see a larger population of our females generating what we would consider resilience. So that's one thing to consider. But then in terms of looking at or working in the field of actions and habits, one always has to consider sex, because there are some really, really nice studies using the 4-core genotype mice from Art Arnold showing that females develop food-reinforced habits more rapidly than males, and this is sex hormone dependent. So it's not gonadal sex dependent. And so that's something we have to consider when we design our studies. And then conversely, Jackie Barker and others have shown that when the reinforcer is alcohol, males develop the habits more rapidly than females. And so certainly, although I think NIH would like us just to dump males and females together and pretend like there are no sex differences when we're writing, writing our grants, we just can't, because the propensity to develop habits is fundamentally different. So I hope that answers your question. Yeah. Good enough. Yeah.
Very convincing data, by the way. Really, really good stuff. I want to ask you about the framing of the findings in terms of habits.
Mm-hmm.
So you probably heard out of this question many times.
Yeah.
It seems from your smile. But it seems to me that there's an informational component here. And in the example that you gave with the glasses, you know that this wouldn't do anything, and you still do it, and that's a habit.
Right.
But if you don't know if that's going to do anything, and you need to learn at the moment.
Right.
So, for example, if it's a broken
light fixture.
something,
Yeah.
you go check if you're going to get the reinforcement, but you don't know at any point whether you should take all the information and the information into account. Can you look at it in terms of information influence?
Yes. So I think the question that you're asking is, when we're talking about habits here, are we talking about habits that result from, say, lots of experience in striatal-dependent action versus habits that are maybe default behaviors that are due to a failure in updating our response strategies because we failed to learn that new response strategies would be warranted? Is that fair? So I think that what we're studying throughout all of this is the latter. So we see that the animals are deferring to what I'm calling a habit-based strategy, and many of us in the field do use that term, even though it doesn't differentiate between the two quote-unquote types of habits, right? So you have overtraining habits, this me with the glasses thing, or this morning I was in the ladies' room and a gentleman walked in. I suspect that was his habit going awry, right? His habit is to turn right, and he was just on the incorrect floor. So that would be a striatal-dependent overtraining habit. Versus a habit by default, where we engage in a familiar behavior because we failed to learn that something different was better. And so I think because that new learning is prefrontal cortical dependent and we're mucking up the prefrontal cortex, that's really what we're studying here. And we can confirm that by taking the mice after the probe test and then continuing to reinforce both nose poke responses and then taking them through another cycle of contingency degradation. And the reason that this helps us quite a bit is that if you have developed a habit through your striatal dependent processes taking over your behavior, then you're going to continue to be insensitive to that contingency degradation because we reinforce that habit by continuing to train you on both nose poke recesses. However, if you just fail to learn that your actions produce consequences, that additional training actually improves your ability to then surmount the contingency degradation procedure. And so in virtually all of the experiments that I've shown you today, we did take those extra steps. We retrain the mice using a fixed ratio schedule of reinforcement for 4, 5, 6 sessions, take them through another cycle of contingency degradation procedures. And in all the cases, in cocaine exposure, in the case of BDNF knockdown, truncated TrkB overexpression, our mice can quote unquote catch up. So I think what we're seeing is failures in prefrontal cortical dependent learning and memory that then cause this deferral to what we call a habit-based behavior. So this language is something that we struggle with in the field. So my colleague Andrew Holmes distinguishes in his studies between early habits and late habits. And I think that sort of maybe imperfectly gets at the idea, but yes, it is. We call these behaviors operationally habits, but you can certainly arrive at these habits through at least 2 different avenues. Yeah, thank you. Yeah.
So the links to structural plasticity
Yeah.
and spines are potentially really interesting. There's some work recently from Josh Wang's lab about connectivity down to VLA
Okay.
and how cells that project down to VLA talk to each other very differently than the ones that don't project.
Oh, cool.
So I was wondering if you have any thoughts on if there's specific cell populations that are really driving all of this. Those are the ones that are experiencing plasticity.
Yeah, right. So that's the question. We've been able to block some of the plasticity that would be associated with new learning using things like latrunculin A, which blocks effect and polymerization. And so we've mostly gone in using pharmacological approaches that are not cell type specific. And so one of the next steps for my lab, and may NIH smile on me, is to start to look at cell type specificity. Yeah, I mean that's essential, right? Like it's the clear next step. Yeah.
This is a pretty naive question, but I started me thinking about habits and the associated cost of the habit, right?
Mm-hmm.
So the example you gave with the glasses, there's no real
Right.
there's no cost to you, there's no real effort it takes and so on. But I think of addiction habits just naively as
Yeah.
like people or animals going to great lengths despite maybe negative consequences.
Yeah.
So can you maybe talk a little bit about that?
Yeah, so another thing that I would really like to do, again, may NIH smile, is look at habits that persist despite negative consequences. And we can do this very easily in rodents by simply equipping our conditioning chambers with foot shocks that deliver shock if the animal continues to seek drug or continues to seek reinforcement. And so a handful of labs in my field do this in the context of a cocaine self-administration that is cued. And so that makes it really difficult to understand whether the cocaine seeking is habit-based because there's an explicit Pavlovian cue. All we would need to do is strip out that cue, and then we can look at whether these cocaine seeking habits indeed are A, habitual, and B, resistant to punishment. Yeah, thank you. None of my tech students? I see you guys.
Is there one more?
No? These guys took a class of mine, so I was wondering if, no? I won't name names. Okay, thanks, guys.